Good morning, folks. So here we go. Today, what we want to do is I want to give you guys an example of how the scientific method works. Each of the steps in a real world example, and we're also going to start talking about some new terms. The New York State Regents always wants you to be able to identify what are constants. What is the independent variable? What is the dependent variable? What is the hypothesis? So those sorts of things are what we're going to work on just a little bit today. We'll continue to practice them, but for now, let's take a look at an example for how an experiment might really be set up. So over here, you see, if you remember, step number 1 of the scientific method is to define a problem. And in this case, the problem is defined. Zach wants to grow tomatoes. He's never grown tomatoes before. And so he really doesn't know what's the best situation, what's the best conditions. How should this be done? So there's his problem. Now, after step number 1, defining the problem, then step number 2 comes in. Think for a moment to see if you can come up with it. Step number 2 is to gather data. So now he needs to go out and try to figure out whatever he can. So in his research about growing the fabulous tomato, he finds out about this stuff called a colored plant strip. And they're a long plastic strip that's designed to go down on the ground, and then you plant through these holes that are provided, and the plant will grow up through that hole, but like the ground and the roots and all that will be covered by this plant strip. So these things are supposed to reduce the water that the plant needs by 30% and increase how fast it grows. And they come in all the colors of the rainbow. So that's the data that he collected. Step number 3. Now he needs to form his hypothesis. Now remember, the only thing important about a hypothesis is, can it be tested? Doesn't matter if it's right or wrong. So he makes a hypothesis. He picks a color at random, green, because plants are green, so green color strips. If I use green colored plant strips, then tomatoes should grow faster. A clear, good hypothesis. What's changing and what he expects to happen. Remember, hypothesis always should be that if-then statement. Specifically, what we're doing and what then we expect to happen. Never a general, never something simple. It always has to be specific. All right, now we set up the experiment. So here's where those new terms come in. So the independent variable, we saw this in question 2 of the airplane lab, where it said, pick one characteristic that you are going to change. This is your independent variable. So a variable is something that changes, and it's independent because it doesn't matter on the rest of the experiment. We're the ones who are changing it. We are the ones who are determining what that variable is. So it is independent of the experiment. So in this case, for Zach's example, he is using the colored plant strips. That is what he is choosing to change. Therefore, it is the independent variable. Now, the independent variable will influence the dependent variable. If someone is dependent on somebody else, then there's a very direct connection between them. The well-being of one person is directly impacted by whoever they are dependent on. So the dependent variable, once again, relies on the independent, will be influenced by the independent. And the key here is that's going to be your measurement. Your dependent variable will be whatever it is you're going to measure. So in this case, the best way to tell if plants grow is to measure the plant's growth, our dependent variable. And I understand if these start to get a little bit confusing, they always do. Don't worry, we've got lots of time. And then finally, the constants. So if we're changing one thing, only one thing, the colored plant strip, we should keep everything else the same as much as humanly possible. And no experiment is perfect. Everybody understands that science exists with a certain margin of error, because we can never keep everything the same for every single test, but we should try. So if Zach was setting up his tomato plants and he put a couple in his living room, one in his bedroom, maybe one in the bathroom, and 4 more in the living room, all of the different colors of CPSs, would that be a good test? Well, no, they're not all the same. It's safe to say the one in the bathroom probably will get a different amount of sunlight than the one in a living room or possibly a bedroom, or maybe the bedroom gets the least. Either way, there's a variation. That's not good. Our constants should be everything else. So if we're changing, let me get the pointer. If we're changing our colored plant strips in order to measure the plant growth, everything else should stay the same. We want the type of pot that each of the tomatoes are in to be the same. We want to make sure they all get the same amount of water, same amount of soil, same amount of light. We want to keep them in the same temperature. We want to make sure that there isn't more wind in one room than another. There are 3 key things that you can keep in mind for this, and these 3 constants will work for almost every single experiment ever. Temperature, light, and water. Those 3 things will work for about 99% of all experiments that in biology anybody does. And on the Regents, they will regularly give you an experiment and then say, what are 2 constants that should be maintained here? Well, that's easy. Take a look at the experiment. If it's testing water, go with the other 2. If it's testing temperature, instead, go with the amount of light and the amount of water. So just flip-flop those 3 however you need in order to make them fit. All right, so we have the experiment, right? He's going to put colored plant strips on his tomato plants. He's going to try to keep all the rest of these things the same, and he lets them grow. Now we need our conclusion. So as we're going towards that, our experimental, remember, is the colored plant strips. We should always have one control. That's what you just saw back here. There's no color on this plastic. So this is our control. It's one natural with none of the experiment stuff done, because if all we did was the experiment, how would we know if it's better than normal? So you always want one normal just to make sure to reference everything back to. So that'll be our control. In the paper airplane lab, your control was one paper airplane that was completely normal and had not been modified. And let's take a look at the results then. Week 1, week 2, week 3. Now, if you take a look at these measurements, do we accept or refuse Zach's hypothesis? He said that if he uses the green colored plant strips, then it will grow more. And you should see that we need to reject that hypothesis, because out of all of these, while the green grew well, it did not grow as well as the control. So it did not grow the best. We can reject his hypothesis, and now we'd go back. So the colored plant strips do not do what they're supposed to. Nice and easy. Now today, I'm going to have you do the gummy bear lab, which will be taking the same sort of an idea and now applying it to some sort of food item. And there's a separate video for that hopefully will help explain it. With any, all right, let me read. With any luck, this helps you to better understand the method, and we will continue to review it as I see you next week.